Okay, ladies and gents, so today we're going to look at the mandatory practical associated with photosynthesis. The SEC will always ask it like this, so they will give you a choice of the influence of light intensity or carbon dioxide concentration on the rate of photosynthesis. I always look at the light intensity, but you can answer the question in either context, and as long as you're getting it right, it doesn't matter. Now, we've already done rate when we did enzymes. So when we did enzymes, the rate has to have a time component. We measured volume of foam per minute. And today we will be measuring something to do with photosynthesis, and again we will have our time component on as a minute. So I'm going to play you the video, experimental video that goes with the practical. I apologize for the sound quality.
In this experiment you have a choice of showing light intensity or carbon dioxide concentration. We'll talk about the light intensity to show the effect of light intensity on the rate of photosynthesis. We're going to use a plant, an underwater plant called Elodea, also in English called pondweed. This is a plant that grows underwater, and it's used to produce oxygen. It's used as an oxygenator in aquariums and things like that because it gives off oxygen. Photosynthesis produces oxygen. It gives off oxygen, and we can see the oxygen bubbles and count the number of bubbles. The more bubbles we get, the greater the rate of photosynthesis. First of all, we take a test tube and we take some of the pond water. Elodea wouldn't be comfortable growing in tap water, so we use pond water. And we cut a sprig of the Elodea. Cut a longer sprig than that. Now, in cutting the Elodea, we cut it at an angle, at a slight angle, and that will give us a better stream of bubbles. And having cut it, we then remove some of the top leaves, just 2 or 3 of the top leaves, just for visibility, so that we can see the bubbles that emerge. Sometimes you have to give a little squeeze to the top of the stem to allow the oxygen to come out. We place the Elodea into the pond water and push it down below the surface. To make sure that the temperature is carried out at a constant, that the experiment is carried out at a constant temperature, we place the Elodea in a water bath. And the ideal temperature for Elodea would be 25 degrees. So using a thermometer, we set up the water bath at 25 degrees centigrade. We then place the Elodea in the water bath and we switch on the light. Now, the plant, we're using a meter rule here to measure the distance from the light source, from the front of the light source to the front of the water bath. So at this point, we're 100 centimetres away. That would represent low light intensity. We have to leave the apparatus set up for 2 minutes to stabilise. In other words, to allow the Elodea to adjust to the new light conditions will take 2 minutes. So after 2 minutes, bubbles are forming. And what we do is we count the number of bubbles produced per minute. And we do that 3 times and get the average. So just counting the bubbles per minute. And we do that 3 times to calculate the average. The results at 100 centimetres were that the average number of bubbles per minute was 6 bubbles. So there were 6 bubbles per minute at 100 centimetres. We now move the apparatus in to the 80 centimetre mark, lining up the front of the water bath with the 80 centimetre mark. And we have to leave the apparatus there for 2 minutes to stabilise. Having left it for 2 minutes to stabilise, we count the number of bubbles per minute. And we do that 3 times and calculate the average. At 80 centimetres, the average number of bubbles per minute was 12. So 12 bubbles per minute at 80 centimetres. We now move the apparatus in to the 60 centimetre mark. Again, lining it up at the front of the water bath with the 60 centimetre mark. And we leave it 2 minutes to stabilise. Having left it for 2 minutes to stabilise, we again count the number of bubbles per minute, and we do that 3 times and calculate the average.
Okay, so you get the idea in terms of how the experiment is set up. Here's one we made earlier, but we're going to start again. So here when we are drawing our experiment, and I'll use the same setup he has done, and I'm going to annotate ours a little differently, or with just a little bit of extra detail. So we are going to have our lamp. We will have our water bath. We are going to put our test tube in here, and we are going to put our pondweed down here, and we are facing it stem up, such that we can now see air bubbles. So in terms of our labels, I'll change the color of the pen. So we are going to count bubbles per minute. So that's going to give us our rate. We have our water bath at 25 degrees. And the reason we're choosing the 25 degrees is because it is the enzyme's optimum temperature. So yesterday when we did the dark stage, we said that there are enzymes involved, so temperature will affect it. So we choose 25 because that's going to be the optimum. We have in here our pondweed or our Elodea, and we have it stem up. And the reason we do this is so that the bubbles of oxygen don't get caught in the leaves. So we have it stem up. And the reason we pick an aquatic plant is that if we were to do this with a terrestrial plant, to try put it underwater to see the bubbles of oxygen, it just wouldn't work. Now the other addition that we make to this, I'm going to change the color of pen. So in the video the gentleman said that they would use some of the pond water for the Elodea. In reality though, in the lab, if we were to do this, I add sodium hydrogen carbonate. So when we do this experiment, we just want the effect of light intensity to be the only variable, the only change that we make every time we take a measurement. So by adding the sodium hydrogen carbonate, we keep the carbon dioxide levels constant. So that gives us one independent variable. So that's going to keep carbon dioxide constant. And we have our water bath to keep our temperature constant. So here is our meter stick for measuring the distance. And this is our lamp. So usually we will move the lamp closer and we will wait. And usually we wait for about 5 minutes. And the reason we're doing this waiting is to allow the plant to get used to the new light intensity. So for the, you now know that the light hits the chlorophyll pigment clusters, they pass that energy to a reaction center. That reaction center passes the energy to electrons, and those high energy electrons get picked up and go pathways 1 or 2. So that takes time to get up to the right speed. So if we were to start counting beginning, we'd have a really low number, and by the time we did our third count, the number would be much higher. So we wait to allow the plant to get used to the light intensity, to adjust to light conditions. Okay, I think now that is pretty much everything that we have in there. So pause if you need to. I'm going to move on. Now, you have this table in your book. The only thing that is incorrect is this. So usually light intensity is 10,000 divided by your distance squared. And in the video, he would have given you numbers of 6, 12, 18, 22, 24. So what it's showing you is as light intensity increases, your rate of photosynthesis increases. The reality is though, for the SEC, they are more interested in the graph. So when we draw our graph, and you've already seen this, when we draw our graph, we will show those results. Now, This virtual lab unfortunately doesn't exist anymore. It was really, really good. I was involved in its development when I was in the UK, but before it went down, I took a recording of it. So there is a link in your assignment, and it just gives you an idea at how fast the bubbles come when the light intensity changes. So that's this, or something very similar, comes from the University of Reading. I'll show you this animation, the top one, very quickly. It will only work if you have an actual Swift player. The thing is called Elmedia if you have an Apple product. And you just need to Google Swift Player for Microsoft or HP product that will work with those. So you could change your temperature if you so wished. So we would obviously do ours at 25 degrees. We would want to make sure we have as much carbon dioxide constant in our setup over here. This little thing will change your light intensity, and you can start your clock, and once you start your clock, bubbles should start appearing and you could count them for yourself. So maybe when we go back into school, if our LODIA doesn't work, because that is a thing that happens with this experiment. But as I say, you have a version of this that you can just have a look at, and it'll give you an idea about what is the actual speed, how many bubbles come out, and how many would you have to count. Now you have these questions. It's the top of page 217 for me. So you should know what changes, what is our independent variable in this experiment. So we're looking at changing the distance between the lamp and the plant. Essentially we're changing the light intensity. The measurement we make is the number of bubbles per minute, so that is our rate. The things that have to stay the same, so the control variables, they are the temperature and the carbon dioxide concentration. And we keep our temperature constant using a water bath, 25 degrees, and we're doing this for optimum enzyme activity. So that is the dark stage part. Okay, so pause if you need to. In terms of how we keep our carbon dioxide levels constant, we're adding that sodium hydrogen carbonate. And the reason we use an aquatic plant is, for starters, it can photosynthesize underwater and it'll produce bubbles of oxygen for counting. So as I say, if we were to do that with basil or coriander or something, it wouldn't work at all. And we position the plant stem up so that the stream of oxygen bubbles can be seen and counted. Now, in terms of our results, they are usually going to be, you can express them as in a table or in a graph, and it would be measurement of light intensity and rate, so it can be a table or a graph. The reality is you will never have to produce data for the SEC, and they usually don't make you calculate it. They do ask you to draw the shape of the graph. So you have this version here, and this is what the graph looks like. So you need to label your axes, and usually when I draw it, I just draw, I don't even do the curve, I just level it off for myself. And it's this point where it levels off. The level off isn't as clear here where it is. You'd need a ruler to work it out. It may level off about here, whereas in my version of the graph it's very, very clear that there is a leveling off. So we have rate, we have light intensity, and what this graph is telling us is that when the light is far away, the light intensity is low. Won't let me change. So this is far away lamp, whereas here the lamp is very close. Now we need to be able to put this into words. So in terms of explaining the result, and I should make the point that this X mark, we call it the saturation point. So Irish people understand the word saturation. If you go out and you get saturated, you're soaking wet. When we're talking about this experiment, the plant is light saturated. It doesn't matter how much more close you put the lamp to it, or how, it won't matter. The rate of photosynthesis will stay the same. So at this point, your electron acceptors are working at their maximum, and nothing you can do is going to change that in terms of light intensity. now, so we'll just go back here and make sure we have it. So in terms of explaining the result in words, we are saying as light intensity increases, your rate of photosynthesis increases. And once you get to that saturation point, so at saturation, increasing the light intensity has no impact on the rate. So it just levels off. Okay, so that's going into page 217. That is the graph shape over here, and this is the explanation over here. So if you are a commercial grower of plants, you want to make sure that what you do is going to make your plants grow bigger, make them grow faster, and where I live on the way home in winter after prep at 10 o'clock at night, there are greenhouses and they will have their lights on. So very quickly, our natural source of light is sunlight, but actually commercial growers will use lamps in greenhouses, and we now know that those lamps may be different colors depending on the color of the leaves. So we've said it'll be the reds, the oranges, possibly the blues, the violets, but they'll use different colors to make sure that you are getting maximum growth. So again, that's our thing, and they'll obviously can only go so far, because once you reach saturation, well, then it doesn't matter what you do after that. So we've already seen this. So you do your lamps to increase your light intensity. It'll only work until saturation is reached, and after that then you're wasting your money on your lights. So in terms of our plants, we know that they'll get carbon dioxide from the atmosphere, and they'll get carbon dioxide from their own respiration, but actually they can use gas burners in a greenhouse, because one of the products of burning gas is carbon dioxide, and that then therefore will increase. So burning gas releases CO2, and that is a way then to have loads of carbon dioxide present for your plants to use it in the dark stage. Now there's a plus side of using burners, and we'll see that in a minute. So your source of water is usually absorbed from the soil by your roots, and we've already done this when we did our plant structure. It'll move up through xylem and it'll be used for photosynthesis in anywhere that's green. Now the video won't work for me here, but this is just showing you all of the things that a commercial gardener could do. They have their light source, they'll have their water source, they can add moisture to the air, they'll have a carbon dioxide source to release gas. They'll also have heaters, and in terms of the heaters, so in terms of your heaters not only produce the CO2 for you, but they will also increase the temperature, which means your enzymes will work faster. So you obviously want it around your optimum for your enzyme activity. And if memory serves, that brings us to the end of the photosynthesis practical.